Okay, hello to all of you. Let's start discussing number 12b. So, in this chapter, we are going to talk in details about the process of endo and the exocytosis. So, I expect you guys already know what endo and exocytosis is. So, basic processes you already know. So, basically, anything, so you know that across the membrane, transport is taking place. Um, Things are diffusing in and out, and some of the things are moving through the transport proteins. We have been talking about all these things, but anything which something is very big, how do they enter and exit the cell? So they some of the things which are very big, um, they enter and they exit the cell with the help of the formation of the vesicles. So anything big exiting the cell, that process is called exocytosis, and anything big entering into the cell, this is called endocytosis, and this takes place with the help of the formation of the vesicles. So, basically, by exocytosis, um, the basically the excretory vesicles, they release the contents outside the cell, and by the process of endocytosis, cells internalize the some of the material which is outside the cell. So this process of the exocytosis, um, it can be of we can divide this excretory pathways by which the molecules are transported to the exterior of the cell. We can actually divide into they can be of three kinds. The secretion pathways can be of three kinds. They can be constitutive, regulated, or they can be polarized secretion. So constitutive secretion is unregulated, is an unregulated process which is continuous, and it is independent of the external signals. And one example of constitutive secretion is the mucus secretion by the intestinal lining. Regulatory secretion is the process which is in response to specific signals. So excretory vesicles involved in regulated secretion accumulate in the cell and fuse with the plasma membrane only in response to specific signals. Example is neurotransmitters, the release of neurotransmitters. And polarized secretion is basically exocytosis of specific proteins which is limited to specific surface of the cell. For example, the intestinal cells, they secrete digestive enzymes only on the side of the cell that faces into the intestine. So this is polarized secretion. So basically various things can be basically animal cells secrete hormones, mucus, milk proteins, and digestive enzymes by the way of by exocytosis. So you already know this process, how the exocytosis takes place. So basically something which has to be secreted outside the cell, it is in the vesicles, and the vesicle moves towards the plasma membrane, and then the vesicle is going to be fused with the plasma membrane, and it's going to deliver its content outside, and the plasma membrane here actually gets extended because of the fusion of the vesicle with the plasma membrane. So the basic process you already know. And on the other hand, endocytosis is basically something from outside, when something big from outside of the cell has to enter into the cell. So in that case, the membrane invaginates, forming a pocket containing macromolecules or other material from the extra. So this is some material which is going to enter inside the cell. This membrane is going to form it's going to invaginate, and basically it's going to form a kind of a vesicle here. It's going to pinch off from here, it forms a vesicle. So the material which was outside the cell now enters into the cells. So the vesicle separates from the plasma membrane, carrying the material from the exterior. So basically in this, what is happening is the membrane, the plasma membrane is actually invaginating, and the material from outside is coming inside, and then this invagination which is formed, it's going to pinch off from here, and this vesicle is going to enter into the site. So basically in the process of endocytosis, what is happening is the plasma membrane is actually losing some membrane from here, because some part of the membrane is actually now forming the vesicle, and it is opposite which is happening in the exocytosis. In the exocytosis, because the vesicle from the inside of the cell comes and fuses with the membrane. So here the plasma membrane gets extended, but the opposite is happening in the endocytosis. So in the endocytosis, the membrane actually becomes some part of the membrane is actually now forming a vesicle. So exocytosis and endocytosis have opposite effects in terms of membrane flow. So exocytosis add lipids and proteins to the plasma membrane, whereas endocytosis removes it. And the steady state composition of the plasma membrane results from the balance between the two processes. So I hope you know, so endocytosis can be of three kinds. It can be phagocytosis, pinocytosis, and receptor-mediated endocytosis. I'm going to focus more on the receptor-mediated endocytosis because it involves different kinds of proteins. We are going to talk about the process, the receptor-mediated endocytosis process in more details. So phagocytosis, you know, when something solid is entering into the cell. Pinocytosis is when some fluid or the liquids are entering into the cell. So I'm going to skip this part. Phagocytosis, you have been studying this from long time. So you can go over that. Phagocytosis in amoeba, and your immune cells are involved in that. You know all these things. So but what we are going to focus on is the receptor-mediated endocytosis, and this is a very important part of this chapter. So receptor-mediated endocytosis, so something which is entering into the cell through the receptors on the membrane. So I'm going to explain you this process with the help of this slide, and I will keep going back and forth so that I can explain you. So here you can see, first let's go here. Yeah. So specific molecules, which we call ligands, bind to the receptors on the outer surface of the cell. So you can see here, these are the receptors, okay? These are the receptors, okay? And these are the ligands. So this is something which has to enter into the cell, okay? So how it is going to enter into the cell? By first of all attaching on these receptors. So these are the receptors on the cell, okay? So these are these Y-shaped structures you see. These are the receptors, and this ligand is going to go and bind to these receptors, and this ligand, when it binds to these receptors, it forms receptor ligand complexes, okay? Okay, so once these complexes are formed, receptor ligand complexes diffuse laterally. So they are going to diffuse laterally. So you can see how the lateral diffusion takes place, okay, and encounter specialized regions called coated pits. And these are the sites for collection and internalization of these complexes. So you can see here, so they will encounter these regions, okay, and these regions are called coated pits because here are some proteins already there. Okay, so receptor ligand complexes diffuse laterally and encounter specialized regions called coated pits, and these are the sites for collection and internalization of these complexes, okay. Accumulation of complexes in the pits trigger the accumulation of additional proteins on the cytosolic surface of the membrane. So here, so once this happens, so once these receptors encounter these regions, these coated pits, so there will be invagination of this whole thing, and there will be recruitment of some more proteins like dynamin protein, and there are some adapter proteins, okay. So accumulation of complexes in the pits trigger the accumulation of additional proteins. These proteins are adapter proteins, clathrin and dynamin. They induce curvature and invagination of the pit, okay. So these proteins, they are going to make this vesicle, they're going to make this this part of the membrane where this ligand and receptor is bound. It's going to have this kind of a curvature, okay. So these proteins induce curvature and invagination of pit, and eventually the pit pinches off, forming a coated vesicle. So eventually this pit is going to pinch off with the help of this protein, and it forms a coated vesicle. Okay, and after this what happens is this coated vesicle becomes uncoated, and all these coat proteins, they are recycled back. Okay, so these are all recycled back. They go back, like leptin, adaptor proteins, they're all removed. These coat proteins, they get recycled and they go back to these regions of the membrane. Now this uncoated vesicle, okay, this uncoated vesicle which we have already studied in lysosomes, this uncoated vesicle is going to be fusing with the secretory vesicles which are going to be coming from the tegion of the Golgi. This we have already talked about in the last chapter, and they are going to form early endosomes, and the early endosomes, they're going to form late endosomes, and eventually they're going to form lysosomes. And then the digestion of the material, the digestion of these ligands will take place, and some of the things, and also during this process, and remember that these ligands, in these vesicles, the ligands are bound to the receptors, and once the this early endosome becomes late endosome, so the pH is going to actually in the development of these endosomes, the pH is going to become very low, and when the pH becomes low, these ligands and these receptors, they're going to be disassociated, and these receptors will go back to the membrane again. They will be recycled, and now what is left in this vesicle, in this early endosome? It's only the ligand, and the ligand, so this early endosome will become then late endosome, and then it will become lysosome. So the material which is there, the ligand which is there, it's going to be digested with the help of the enzymes in this vesicle. And again, these vesicles, they can also secrete their enzymes, their hydrolytic enzymes outside the cell. So this is the way basically how the receptor mediated endocytosis takes place. Yeah, this is what I have just explained. This is what is happening here, and yeah. Yeah, let me explain one more time this whole thing. So basically, these are the receptors on the membrane. This ligand will first of all bind to these receptors, and they form receptor ligand complexes, and there will be lateral diffusion of these ligand and receptor complexes will take place, and they will encounter the regions of the membrane which have some proteins here on this side, and this is called coated pits, and this will lead to the recruitment of more proteins like adaptor proteins, dynamin, clathrin. So all these proteins will help in the formation of curvature and the invagination, and this vesicle is eventually going to pinch off, and it will form a vesicle like this. And because it has all these proteins on this side, these are called coated vesicles. And then this uncoating will take place, and all these proteins, they will be recycled back. They will go to these regions in the membrane, and then this uncoated vesicle is going to fuse with the it's going to be fusing with the early it's going to be fusing basically with the secretory vesicles which are coming from the Golgi, and those vesicles have lysosomal enzymes in them, and these uncoated vesicles and the secretory vesicles from Golgi, they fuse together and form early endosomes, and the pH in the early endosomes will decrease, and these ligands and the receptors, they will disassociate, and these receptors will be recycled back, and then these early endosome, they will be their pH will decrease more, because these vesicles, they have proton pumps in them which keep on pumping protons inside, because of which the pH becomes low. Then the early endosome will be converted into late endosome, and then from the late endosome they will be converted into lysosomes, and the digestion of the ligands will take place. So this is the process of receptor mediated endocytosis. Yeah, that's basically what I have been just written here. And clathrin independent endocytosis, basically fluid phase endocytosis, is a type of pinocytosis for non-specific internalization of the extracellular fluid. And this process does not concentrate the ingested material just the way it is taking place in the receptor mediated, because in that the material is concentrated because the ligands bind on the receptors. This does not concentrate the ingested material, and contents are routed to early endosomes. Okay, now we are going to talk about, so we just talked about overall the process of receptor mediated endocytosis, and we talked about the coated pits and the coated vesicles. So now we're going to talk about specifically about these proteins which are there in these coated vesicles. So so let's talk about these proteins. So there are several type of coat proteins, and the most studied coat proteins are clathrin, coat protein 1 and coat protein 2. These are the most important coat proteins. You need to know their names. The type of coat protein on a vesicle, okay, the type of coat protein on a vesicle helps to determine the destination of the vesicle. So we basically right now talked about, just now we talked about the receptor mediated endocytosis. Now there are, like, in this whole process, you know, all the endomembrane system is actually connected through these vesicles. So we are now going to talk about other different kinds of vesicles and what kind of proteins they have. So here you can see these are the different kinds of coated vesicles. So they can be clathrin coated, they can be coated with coat protein 1 or with coat protein 2, and these are the specific proteins which are there there in the coated vesicles. Like if the vesicle is clathrin coated, it can has coat proteins as clathrin, it can has it has adaptoprotein 1, ARF designates the full name is given here, it's ADP ribosylation factor 1. Then it's, so now here if you see in this table, the origin and the destination is also given here. Okay, so we are talking about coated vesicles which are found in eukaryotic cells. So this kind of vesicle, clathrin coated, they can originate from the trans-Golgi network of the Golgi, and their destination can be endosomes. And this is another clathrin coated protein, but the coat proteins in them are different. They can be clathrin, they can, in their coat, the coat proteins can be clathrin or they can be adaptoprotein 2. So their origin can be from plasma membrane, and they can be, the destination can be endosomes. So just remember that from where they are originating and where they are, what is the destination. So these kind of vesicles both are clathrin coated, but these guys originate from Golgi and they go to endosomes and they form the endosomes eventually. And these ones, they originate from plasma membrane and they go to endosomes. Now coated vesicle with the coat protein 1, these are the coat proteins. So these originate from Golgi and they go back to either Golgi or ER. Okay, so these are the vesicles which originate from Golgi. They can go back to Golgi or they can go back to ER. Okay, and coat protein 2 vesicles, coated vesicles, they have these proteins in them. They originate from ER and they go to Golgi. So these are all basically coated vesicles, and this table basically shows from where they originate and where they can go. What's the destination? Okay. Okay. So now we are going to talk in more detail about the protein clathrin. Okay, so we are going to talk about the basic unit of the clathrin and how does it helps in the formation of the curvature in the vesicles. So clathrin coated vesicles are surrounded by coats made of two multimeric proteins, clathrin and adaptor proteins. Okay. Now the shape of clathrin proteins and the way they assemble, so they are multimeric proteins, which means they're made of more than one polypeptide chain. The shape of clathrin proteins and the way they assemble provides the driving force to induce a flat membrane to form a spherical vesicle. So now you will understand that what change takes place in this protein, in the basic structure of this protein, which makes this vesicle to get the curvature. Now the basic unit of clathrin is called triskelion. Okay. Now what is the structure of triskelion? So this is the basic unit of clathrin. So this is triskelion. Now this is made of 3 heavy chains and 3 light chains. So you can see how the structure is. So this is the heavy chain, okay, and these are the light chains, and they can form shapes like hexagon or pentagon. So this is the basic structure is triskelion, and it can assemble in different ways. It can assemble in the form of hexagon or the pentagon. Okay, so triskelion, each is a multimeric protein composed of 3 heavy chains and 3 light chains. As I said, these radiate from a central vertex with the light chains associated with the inner half of each leg. Now, as I said, triskelions assemble into hexagons and pentagons of the lattice around clathrin coated proteins and vesicles. And also in these clathrin coated vesicles, there's another protein involved, is the adaptor proteins, and they have the ability to promote assembly of clathrin coats around the vesicles. So they basically promote the assembly of clathrin coats around the vesicles. Now something very important to know here is the assembly of clathrin coat, the assembly. Okay, remember that clathrin is, its basic unit is triskelion. Triskelion can form hexagons or it can form pentagons. So the assembly of clathrin coat appears to provide some of the driving force for the vesicle formation. Okay, initially all clathrin units are hexagonal and form a planar structures. So when the clathrin units are hexagonal, they form planar structures. As more triskelions are incorporated into growing lattice, some pentagons are also formed. Okay, the mix of pentagons and hexagons allow the new coat to curve around the budding vesicle. So the curvature which is formed in the vesicle is like spherical. So it is all because of the incorporation of the pentagons in these hexagons. So the mixture of pentagons and hexagons is responsible. It is formed in the vesicle, and spherical vesicles they are formed. Otherwise, if they are only hexagon, then they are planar structures. So you can see here. So some are pentagons, some are pentagons, some are hexagon. So that is why they have this kind of a structure. And then there is involvement of another protein. This we studied in the earlier part also, the protein called dynamin. The function of this dynamin is required for constricting and closing the vesicle. What is dynamin? It's a cytosolic GTPase. Now what are GTPases? GTPases are those proteins. We'll be talking about GTPases more when we talk chapter number 23 on signal transduction, which we'll be talking about next week. So GTPases are those proteins which can convert GDP into GTP, GTP into GDP, guanosine triphosphate to guanosine diphosphate. Okay, so dynamin is basically a GTPase. So as GTP is hydrolyzed, because its function is to hydrolyze GTP into GDP, dynamin ring tightens and separates the vesicle from the plasma membrane, and that's what dynamin protein is doing. Okay, and this basically talks about so we just talked about the clathrin coated vesicles. Next we are moving towards the coat protein 1 and protein coated vesicles. And these, so we have studied in the table here. You can see the coat protein 1 vesicles, they origin from Golgi and they go to ER, and the coat protein 2, they originate from ER and they go to Golgi. So we are now talking about some of the proteins which are involved in these kind of vesicles. So coat protein 1 and coat protein 2 coated vesicles travel between ER and Golgi complex cisternes. Now the coat protein 1 coated vesicles are found in all eukaryotic cells and are involved in retrograde transfer from Golgi to ER. And these are some of the proteins which are there along with the coat protein 1. These vesicles are coated with coat protein 1 and ADP ribosylation factor. So you have to just remember these names. And yeah, I think the most important thing to know for you guys is like the coat protein 1 coated vesicles, what is, from where do they originate. So these guys, these vesicles, coat protein 1, they originate from Golgi and they go back to ER. So they are involved in retrograde transport. So I think that's the most important thing to know. And coat protein 2 coated vesicles, they actually originate in ER and they go to Golgi, and it's all some more proteins which are involved in the formation of these vesicles, which the list is there. And the last thing in this chapter which we need to know is about the proteins which are important in mediating the fusion between the vesicles and the target membranes. Okay, so what determines that the vesicle is actually going to the correct place and fusing with the correct membrane? So for that fusion are responsible some proteins, they are called snare proteins. Okay, so once vesicle is formed, so right now we just talked about the vesicles, the vesicles originating in different parts of the cell, they have different kinds of coats in them. So we just talked about that. Now which proteins determine, once the vesicle is formed, that they are delivered to the correct destination? So those proteins are called snare proteins. And there are names of some in this process. There are some more proteins involved. They're called NSF, that's N-ethyl malamide transfer, and another are called SNAPs. They are soluble NSF attachment proteins. So so these snares, there are two kinds of snares. One is V snare. This is a protein which is present on the vesicle, easy to remember from V. T snares are the proteins, snare proteins, which are found on the target membrane. So now let's learn from this slide actually. So basically you can see this is the vesicle and this is the V snare. Okay, then there is another protein involved associated with this vesicle. It's called RAB-GTPase. Okay, so this is the vesicle and this is your target membrane. So the vesicle has a V snare and your target membrane has a T snare. And there are some tethering proteins which are there on the membrane. The proper vesicle is recognized and bound by particular membrane anchored tethering proteins. So these are the tethering proteins. So now this vesicle will be recognized and bound to a particular membrane by these proteins. So you can see that the vesicle has reached here and these proteins, these are helping this protein to be bound. It's basically helping in these tethering proteins are actually helping in the binding of this vesicle to the membrane. And how it is going to help? By mediating the attachment of this V snare to the T snare. A RAB-GTPase bound to the incoming vesicle stimulates the association of V snare and the T snare. Now this protein RAB-GTPase, this one, is actually going to help more in the association of the V snare and the T snare. You can see that now here this V snare and the T snare, how they are associated, how they have basically associated here. You can see that. And then once they have associated, you can see the vesicle can deliver its material. Suppose this is a plasma membrane and this is a vesicle which is bringing some hormone or something, and that hormones needs to get it out. Now you can see how these V snare and the T snare, they were associated, and now the membranes of the vesicle and the target membrane, it fused and it has delivered the material outside. So binding of, then as I said, there are more proteins involved. Binding of NSF and SNAPs promotes the dissociation. So once the function is done, the work is done, the secretion is done, then these proteins are going to help in the dissociation of the snare complex, and then the vesicle is going to be so this whole complex will be disassociated. So this is basically what it is, that how the vesicles, they reach their destination. So all from this chapter. If you have any question, please let me know.